All right, I gotta go. What we have heretofore discussed in this lecture is talked a little about spermiogenesis, spermatogenesis. We're going to wrap up here with the male stuff. It won't take very long. Then we've got quite a bit to talk about with female physiology. But remember, what's controlling all of this are tropic level hormones involved. First of all, gonadotropin-releasing hormone is going to be the one from the hypothalamus. That's going to go into the anterior pituitary. And in the anterior pituitary, we've got LH and FSH in both sexes. Okay, luteinizing hormone and follicle-stimulating hormone. They're both going to be involved in the sex cell, or in the gonadal region, anyways. Okay, now remember that in the case of males, and in the case of spermiogenesis and spermatogenesis and all that process, we've got two cells that are of interest to us: Leydig cells and Sertoli cells. Remember that the Leydig cells are actually interstitial cells. They are not part of the seminiferous tubules. Instead, they are cells around that are going to be secreting testosterone. And remember that there is just like in the brain, there's a blood-brain barrier, there's a blood-testes barrier as well. So within that, there is no blood flow. It's highly controlled. Okay. Luteinizing hormone is going to act on the hormone production of this region, the testosterone. You will also find that when we get into female reproductive physiology, not Leydig cells, obviously not those, but luteinizing hormone will also have to do with estrogen, progesterone levels, so the sex hormone kind of thing. Whereas the follicle-stimulating hormone, in the case of males with Sertoli cells, axon Sertoli cells, which are where that division of sperm was taking place, that blue-colored slide that we had. If we go back a couple, this guy somewhere in here. These would be the seminiferous tubules, would be the lumen of it would be in here. These are Sertoli cells where this is taking place. Around this, not part of the particular, this is just a little piece of the tubule. All these will be lined up next to each other to form that tubule. Outside of this tubule will be the Leydig cells that are secreting testosterone. Okay. Oh, whoa, I'm going way the wrong way. Okay. Now, when we get into females, we're going to be talking about follicle-stimulating hormone will start taking sort of a primitive version of an egg and cause it to divide, okay, and start making little egg follicles, sort of like you're making sperm here. So there's the difference between those. Okay, I would show you this animation, but we've already seen it. You can look at it yourself. All it shows is this stuff dropping down from the hypothalamus, interacting with these, causing this to be produced, these two to be produced from the anterior pituitary, and then those two going out to the gonadal region and acting on those cells. So it's just another example of that hierarchy. You can click on that yourself. I won't do that here. Realize it's all negative feedback. Look, whenever testosterone is produced, this is signaling for testosterone. Okay? Whenever it starts to be produced, it's going to feed back to the hypothalamus, and when the levels of testosterone increase, it's going to decrease this process. It's going to shut this down. Okay? Also, inhibin is secreted from Sertoli cells, those cells that make the sperm that are in those tubules. That's a hormone that can be released that can also go back and inhibit follicle-stimulating hormone. So the products of these end up shutting them down whenever the product's level gets high enough. Okay. All right, we know this. As far as testosterone, not only will it have an effect on secondary sex characteristics, but it can also play on mitosis and meiosis of germ cells, which are going to be sort of these primitive cells are going to turn into other things, like in the case of spermatogenesis. So, again, follicle-stimulating hormone thus is going to have a huge impact, since that's actually the main driving force behind spermatid remodeling, that whole packaging, the tail, the mid-piece, the head, putting that all together, and that kind of thing. So, estrogen probably plays a role in male health also. It's been studied, although nothing has actually come about of it. I'm not sure the extent to which estrogen plays a role and how it might negatively impact the male body if things get out of whack in terms of estrogen. The gonadotropin, well, other than the obvious things that I'm talking about, like normal physiological response, not taking it, in addition to intentionally, that kind of stuff. We know what can happen, but gonadotropin releasing hormone activity increases at puberty. Why does this growth spurt happen? Why do the gonads develop, turn into reproductively functional at that point? What actually increases in terms of secretion is this, the very first one, the one that comes from the hypothalamus, the gonadotropin-releasing hormone. This activity increases. It starts to, it's not necessarily, it does create a little bit more than what was normally there, but that's not the key. The key is this part. The hypothalamus becomes less sensitive to feedback inhibition of testosterone. So in the case of a male, what happens is during this pubertal growth spurt, and as this is setting in, yes, gonadotropin is being released, but what happens through another series of largely non-understood events is that the testosterone that normally feeds back and shuts this off, this is now becoming basically desensitized to that testosterone. So as normal levels of testosterone increase, normally that would come back and shut that off, and it would maintain consistency, and nothing would change in terms of excessive development and with all this stuff. But instead, during puberty, what happens is this testosterone starts to increase, and instead, it goes back like it should, but instead of negatively feeding back and shutting this off, it starts to require more and more testosterone for it to shut off. That's basically what happens. So it continues, and the overall effect is an accumulation of more testosterone in the body, and then that leads into these changes in this growth spurt stuff and development of the gonads. All right, and there's a flow chart of what I just told you. Okay, now as far as a little bit of anatomy, we have to touch on a little bit of anatomy here. It's just as far as male reproductive anatomy and physiology, it's just a lot of winding tubes in the case of the male. A lot of tubule space down in the actual testes themselves, the seminiferous tubules, and that we've already talked about. There's a lot of winding tubule space there, but there's also at different points feeding into these tubes several accessory glands. One is very common. We hear the prostate gland, but also there's bulbourethral glands. There's some other things, and they all add a different component to eventually what's going to become the semen. All right, so as far as, I mean, you probably know most of all this anyways, but we've already talked about this. The seminiferous tubules are in here. The epididymis is right around the outside, and what you're going to get here is you're already going to have the mobilization and the production of the sperm, and they've already sort of matured. But what's going to happen is they're going to move to those seminiferous tubules, and right here is where they're going to get concentrated, and they're going to get finalized and developed and matured and packaged and that kind of thing. Okay? So that's the epididymis. And then that's going to serve as the exit from the testes, but then we're going to go up through the ductus deferens, or the vas deferens. Okay? You said they're matured in epididymis? Yes. Okay, and then obviously the route commonly known, I mean, we're going to go up through the vas deferens. And what will happen is this is one side, the other side, it comes back on either side of the bladder and sort of V's in the back. Okay, they'll come together. Yep, you should have seen that anyways. Now, I talked about this already, I think, in terms of keeping it cooler, a little bit cooler of a temperature in order to manufacture and to make sperm. This right here, where you're passing through, there's a canal right here called the inguinal canal, which is a passageway from the abdomen out through the body towards where the testes are. There are some, I think you had to learn the cremaster pouch and things like that. That was like a connective tissue that was holding all this together. But there are muscles associated with this. You know, we've mentioned that already. Let's see. I told you here, like I say, in the epididymis, that's where you gain movement, fertilization, the ability of fertilization, the ability for movement. In other words, mature and become functional. Okay, I mentioned this already. Oh, now this picture is a little bit better from the back if we're looking at them this way. I told you they come in and they actually V right behind the bladder. And right where they actually meet, where you start forming the ejaculatory duct, you're going to find these seminal vesicles on both sides. Okay, one feeding into both, and then right at the base of that, where the prostate gland actually starts, is where you're going to actually merge and form one duct at that point. Excuse me. I think I was going to talk about what these things do here in a second. Just making sure. Okay. And then the last gland that we'll mention here is a very small bulbourethral gland on both sides. The next couple slides should tell us what is being excreted by these things and why they're necessary. Okay. Of course, these are feeds in from the kidney, right? The ureters that are coming in from the kidney to feed into the bladder. And then, of course, the biggest difference between what's going on here in terms of the path that's followed involving the bladder and the urethra is that in males, they actually merge, right? The reproductive tract and the actual emptying of the bladder, they share the same path at the end. The urethra becomes used for both functions. All right. Now, as far as what is produced by these accessory glands, now we know that the sperm itself is what's most important out of all of this, right? It's carrying that genetic material to perpetuate our species and our genes. Do what? Well, the egg has all that other junk, but as far as that important key missing chromosomal element there, got to have that. Come on, I got to make myself feel good up here. Just feed me a little, feed my ego a little bit. All right. So what's the point of all these other glands? Well, you know, what is the point of the prostate gland? What's the point of these bulbourethral glands, these seminal vesicles? They have to serve some function, and they do. You know, the semen, of course, is a mixture of a lot of things. You're going to get fructose in there, which is a sugar source. There's going to be some other things that are going to be able to make the sperm survive. You know, the vagina is a very acidic environment. So what's actually part of semen is like an alkaline secretion, like a basic type of secretion that when it's in there can help neutralize the acidity from the vagina and give these sperm a better chance, so to speak, of making it. I'll show you some interesting stats about how many sperm survive at given points. It's kind of mind-boggling. Okay, so these seminal vesicles, so these guys way up here, right? As soon as the sperm are traveling out and heading towards the ejaculatory duct down here, what'll happen is this fluid, from the first fluid from the seminal vesicle, supplies fructose, which is an energy source, a sugar. Not that it's the same, but like fructose, like your Pop's high fructose corn syrup, right? So fructose is a simple sugar. For energy, and prostaglandins. Now, prostaglandins are an interesting group of chemicals because they are widespread throughout the body, and they have many different functions, and they're not entirely understood how they have the functions that they have and what all they are involved in. For example, you take Tylenol because, or the reason that it works is because it inhibits the synthesis of prostaglandins. So prostaglandins are actually involved somehow in the nerve transmission, but it's also involved in pain receptors in the initiation of the pain reflex. So these are molecules that are important in a lot of different things. But what we can do is, like, if you could suppress their manufacturing, one thing that can come out of that is less of it around to be involved with the pain reflex. So that's just one example. But in this case, it can also induce smooth muscle contraction. So you could obviously see why that would be important. Not only for delivering out of the male, but also once in the female, if there's prostaglandins there, that could help initiate movement in terms of smooth muscle contraction. Okay, so these all can work together. Also, up until this point, what's coming up through the vas deferens is actually pretty much mostly just sperm, not the additives yet. So as a result, it's going to be a little thicker, and it's going to need to be diluted a little bit if you're going to be able to move this across the distance. So this is where you're going to find this dilution. It'll add a little bit of a water-based solution to dilute this mass a little bit, but also you're going to add fibrinogen. Where do you find fibrinogen? Where have you heard of that before? Yeah, it's like involved with blood clotting, right? So what will happen here is fibrinogen will be into this mixture, and then once released, it'll allow the sperm to then sort of coagulate, or the semen, I guess, to coagulate and sort of stay together in a general area. Okay, a little bit later. All right, I already mentioned this. These are fatty acid derivatives in terms of how these molecules came about. These prostaglandins, they're modified fatty acids that went through a couple chemical changes. Here's some things. I told you that they're sort of widespread. Here's some things where you're going to find prostaglandins involved with. Obviously, we talked about promoting sperm transport, but also bronchodilation, renal blood flow, acid secretions in the stomach, all these things. So prostaglandins are sort of everywhere. All right, the prostate gland. What is its most important function? I mentioned a minute ago alkalinity is something that we need to introduce into the semen because of the acidic conditions in which the sperm is about to approach. Okay, so you're going to get this secretion from the prostate gland. It'll help neutralize the situation. Also, a little bit more clotting enzymes that can then add to the effect of the fibrinogen up here. Okay, and the bulbourethral glands, those are at the very bottom, down here. Oops. Mucus secretion for lubrication and to enable travel a little bit better. This is a long way to go for a little guy. Okay, not only you start way down in here at the cellular level. Think of how concentrated this is. What's in here compared to what all's happening in here? It all just gets concentrated together, and there's millions, hundreds of millions, literally more than that. A whole bunch, because hundreds of millions actually get ejaculated, and there's even more left behind. So there's tons and tons and tons of these little guys, and they got to make their way all the way out here. Not only that's just getting started. The real work starts after they leave here, and they have to go fertilize that egg. Okay, the quest begins. This is just an initiation process. Once they get there, then it's the work's just begun. It's amazing. If you get into this too much deeper than what we're going to go, and look at some of the biochemistry that's associated with this, and you start looking at what are the odds that this could ever happen in nature, it's mind-boggling. It's just awesome. It's a neat thing. All right, so female physiology, a little bit more complicated. Okay, you'd be surprised. What we're going to focus on is this ridiculous fluctuation. I'm just kidding. Biologically amazing fluctuation of hormones, especially the two. Okay, we're gonna be looking at, as tropically speaking, of course, LH and FSH again, estrogen, progesterone. Those are the big players on this. What we will see, hopefully. Now I know that I'm preaching to the choir in terms of a lot of this stuff, but I'm anticipating that you don't know some of the details that I'm going to talk about, because some of this is it's very interesting when you superimpose and lay everything over the top of each other and put it all together, I guess, and see how it interplays. It's kind of neat. Okay. Well, we know that ovaries. This is easy. We know the ovaries produce ova, which are the eggs, and also going to be secreting estrogen and progesterone. Okay. Now remember, estrogen also can be released from your adrenal glands, right? But for you guys, where you're at, the bulk of your estrogen is coming from is from the ovaries. Now, as far as let's focus on estrogen for a second. What is the role? Obviously, maturation, maintenance of the reproductive tract. That's a biggie. That's obvious. Secondary sexual characteristics, breast development, all that kind of stuff that goes along with it. Ova maturation and release, which is a really, really neat thing, biologically speaking. As if you had anything to do with it, right? Well, thanks. Biology took care of that for you. You get no credit for it. All right. Oh, I thought you said thank you. And I was going to I thought you were trying to applaud yourself. Okay. Well, see, that's just the male genes. Practice. Okay. And then, what people don't realize, I want to throw something in here a little bit later that's actually kind of cool. But I'll just throw a teaser in now. Transport of sperm from the vagina to the site of fertilization in the oviduct. Now, there is some chemotaxis that happens here. Okay? There is some level of communication, as strange as it seems. There is a level of communication between that egg way up here in the oviduct and the sperm that's coming in to try to meet it. Now, I'm not saying it's a one-on-one. It's like it's like a match that was meant to be, and there's this one. Maybe it is. I'm just saying I'm not saying that. that there's one down here and one down here, and if they had eyes, their eyes met, and they met each other. You got what I need. But you say you're just a friend. But no, it's whoever gets there. But there is a level of chemical communication. Okay.
I'm just totally exhausted from this class.
Trying to plow forward. I'm trying.
Hey, we stuck it out the whole semester.
All right, keep going. There's a level of chemical communication, and I want to mention something a little bit later. If I forget, remind me. But there's actually been a couple chemicals that have been isolated that's very interesting that seems to be present only during this time and causes, it seems to have some relationship between drawing the sperm towards the egg. And it's a scent that is actually something you sort of, but I mean an actual scent that you probably have smelled before. Okay, yeah, weird. Okay, I'll explain a little bit later. All right. Now, of course, the main function of progesterone has to do with creating a suitable environment for implantation and nourishing an embryo. Okay, we'll discuss this a little bit further, but they're both equally important. Okay, one thing I know you know all this, but one thing that is interesting is that over here at the end of the oviduct and where the ovaries are, these are actually floating sort of independent of one another. That's neat because there's no surrounding of this and no way to guarantee that you're going to swoop in the eggs that are released. Okay, so when these sometimes you can literally think of it this way. Sometimes the egg could be released way over here, and it has to actually get swept in by all of this to even enter the oviduct, and there's no guarantee that it will. So that's sort of interesting. As far as the anatomy, I'm assuming you guys already know this, but we got the oviduct, the myometrium, which is going to be the thick muscle part of the uterine lining. The uterus. The endometrium would be the uteral lining. Okay, the uterine lining. The cervical canal, right at the beginning of the cervix, and then the vagina up till that point. So that's as far as anatomy goes. We're going to focus more on the physiology. But all kinds of stuff physiologically happening in here on a regular cycle, but also during pregnancy. Really cool stuff. So, all right. Now, as far as to get the hard stuff out of the way, the oogenesis, the beginning of an egg cell. How does that come about? Because we know that it's a little different than in males because, like you have mentioned before, you're born with a certain capacity in terms of egg cells, right? Where males constantly make sperm. What will happen is, I'm just going to sort of skip over this and go to this picture, okay? And then because this is a picture of the explanation. What happens is before birth, you have this first primitive cell called the oogonium, which is sort of like a spermatogonium. It's going to give birth to everything else. All right, so notice the difference here. It says mitotic proliferation. Mitotic proliferation prior to birth. The cells that are dividing are doing so to make just copies of themselves. They are not undergoing meiosis. They are not halving their chromosomes at this point. They're just making more of themselves. Okay, and what will happen is as this is happening upon birth and development, you've got a proliferation in the sense of you've got a meiotic division that this starts then, these ones that are made, they start to divide. And when they start to divide, they undergo meiosis and they halve their chromosome numbers. And what you get actually stored and collected are these guys in your ovaries. Okay? These are primary oocytes. These are not eggs. These are things that have the potential to develop into eggs. Okay? These are primary oocytes that have been arrested in terms of meiotic division. They've started, they've separated, and they've stopped. And there's nothing now. It's really interesting because something triggers them later to continue and pick up where they left off. But right now, they're just sitting there and are in sort of arrested development, so to speak. Okay? Okay. So that's what this is about. Now it's neat because it says the first meiotic division of an oogonium produces a primary oocyte diploid, which is what I just showed you in that first line. It is surrounded by a primary follicle. See that little red line around those blue dots? That's what we're going to call follicle. This is where the whole follicle thing, I'm talking about the hormone and all that. This is why we're going to refer to that. There's actually a little tiny, I'll show you a little bit later, where this turns into an actual layer of cells around the outside. But anyways, you've got this follicle. And it says only about 400, this area right here in the egg cell itself, all of this comprises what we call a follicle. Okay? All right, so it says only about 400 follicles will mature further to possibly release ova in the actual course of life. Now that's just an average, of course, but what that's saying is, you've got a whole lot of this to start with, and you've got the bank of all of these. Okay? Now once that proliferation happens prior to birth. So what you have is an inborn genetic capability to produce a certain number, and you are limited by ever how many of these you have whenever you're born, whenever you get here, and when this stops. Now once this stops and you've got this, that's what you have to work with. And then throughout life, I know that you may not actually go through 400 separate ovulations, but realize that some of these follicles, every time an ovulation happens, okay, to get to that point, one of these follicles had to have matured and started growing into the actual egg. Anywhere from 6 to 12 of those guys will do it at the same time, and only one of them usually gets released from the ovary. The other ones just degenerate. Okay, so that's how come you can still get more than that's how come you can still get this high of a number. So only about 400 of these will mature further to possibly release ova. Some of them will never even get called on. Okay, some of them you'll just stay there. Okay, so most of them will never ovulate, and those able to ovulate may be arrested for years. So it's a lot of biochemistry that goes into this. That's, again, largely it's an area that's not well understood. There's a lot of research out there going on with this. Okay, so like I said, from puberty to menopause, that's what's going to happen. These primary follicles and oocytes are going to mature into secondary follicles and oocytes, which you're going to see down here. In this process, it goes from the bank, and anywhere from 6 to 12 of these guys turn into what's called an enlarged primary oocyte. And one of those is going to end up being the one that actually gets released. Okay, so here's this second primary oocyte. Sorry, that's not true. That's him, the one that, or her, if you want to assign a sex to it. No, this one is just this one right here. It's not divided anymore. It's just swelled up because it's starting to mature. It's going to be one of the ones that get called on. And then when that divides, I'm going to mention this in a second over here, but here's your secondary oocyte. That's just this guy, girl, swelled up, made this, it just become enlarged. It's getting ready to go through this maturation process, and when it does, it divides. Okay? Now this is meiosis. This is why this had to happen. Remember, these are mitotic. These have all your chromosomes present. So technically, remember I told you that some animals have the ability to just make copies of their eggs and get going? There exists in your primary oocytes, you can have cells that are full chromosomal number. So if we could figure out a way to take a woman's primary oocytes and initiate that process of cellular division and cause it to develop as opposed to just go into meiosis, but instead continue mitosis and develop into an embryo, then you could trigger this for development, okay, which is kind of interesting. Now, down here, what we have is what's called a polar body, first polar body. Pole because poles means opposite ends. What happens is this guy has to divide. Now, we've got chromosomes that we have to now deal with during this section. What is happening here? So what will happen is it's got to undergo some sort of change so that we can get down to 23 chromosomes so that we can go down to it can be fertilized with a sperm. So what happens is it splits apart and it undergoes meiosis. So one of them got called on, actually several of them, but in this case we're looking at this one. It enlarged and it said, okay, if I'm going to be used, I've got to split down to half my size in terms of chromosomes. So that's what you get. And this is this secondary oocyte that's been halved. Now what happened to this guy? Well, this is interesting because these are called polar bodies, and these polar bodies don't really seem to serve a purpose. My question is it just to get rid of genetic material? Because biologically that seems what tends to be happening. This is just a way for us to dispose and separate out unwanted genetic material. Now, is there a way to ever trigger this? What you'll see is that this steals all the cytoplasm, and this is very little cytoplasm and just the other half of the chromosomes. Is there a biological function for this? Can we use it for something else? Is it fair to test this, you know, can you use this for tests if you can identify this as opposed to this and that kind of stuff, because technically it's the other half of the, I don't know. But anyway, it's interesting. And what will happen, what's even more interesting is that that doesn't just stop. It sort of stays on track, and how this one then divides again. When this becomes fertilized, okay, and the sperm hits this, what will happen is immediately before the sperm makes it in, it'll trigger another division. And this guy will pull out, and this one will become another polar body right here. So you end up with 3 total polar bodies, because the second that becomes that way and starts to go out, this one divides as well, although probably before this one gets fertilized, but still that pathway is still rolling. This one divides as well. What's the point? Why would it do that? I don't really know, but it seems to divide once more to give you 2 polar bodies instead of 1, both of which are non-functional. We don't use them for, well, that we know of. So they degenerate. They're just a way to do some house cleaning in terms of your chromosomes. Okay, I think I explained most of that, but it's that's right. Well, this is just one cell. This one right here. Right. When this one right here, that's why we don't like make 2 and then end up with, like a sperm, we would end up with 4 sperm here. But in the egg, we end up with 1. It's a more selective process, and that's why you're actually born with only, when they say born with all you're going to have, because each one of those now don't have the ability, like in sperm, to keep splitting, to keep meiotically producing more. Instead, whatever is split is trash. It gets taken out. Okay? So you go from a primary, when that divides, to actually just 1 secondary, and then what will happen is when that gets fertilized, starts to get fertilized, it'll actually split again, and this is going to be the chemical changes that occur between here and here mature that egg, and then before the chromosomal fusion happens, it kicks out another polar body over here. So this all gets taken out as trash. Okay? And you're right, that's a big difference, because in sperm, we get as much as we can. We make as much as we can. With eggs, it's a much more selective weeding out process. It's more of a prize, so to speak. Okay. I think that covered everything there. Okay, so as far as the ovarian cycle, now we're going to look at 2 different things. We're going to look at the ovarian cycle, what is happening hormonally in the ovaries, but also corresponding into the uterus. You know, what changes are not only happen hormonally in the ovaries, but how does that affect what's happening in the uterus as well? And they're obviously tied very closely together. So what we have is alternating follicular and luteal phases. Okay, so we're going to have follicle stimulating hormone, and we're going to have luteinizing hormone that are going to create an alternation in terms of secretion in their patterns and drive this cycle. Okay? All right, so what we have here is, here's your example of a collection of primary oocytes. Now remember, that's these guys up here. Okay? So you have a collection of primary oocytes, and what you have is, these are large, right? We know that we can actually see the human egg cell with the naked eye. We don't need to get a good look at it, you got to use a microscope. But if I dropped a bacterial cell in front of you, you can't even see it. But if I drop a human egg cell in front of you, you could actually see the speck. So it's actually a fairly large cell. So large that these are actually separate cells around it. Okay? These are tiny little, what's called granulosal cells. We're going to see the importance of those in a minute. But they help make this follicle. Okay? These together, all this is going to make the follicle. So here's the primary oocytes. We say the follicular phase is dominated by maturing follicles. What you're going to see is, let's say from day 0 to day 14, then we'll go from day 14 to day 28, okay, just on average. If we do it that way, you'll see that the follicular phase is the first part, okay? And what is happening during that first part is follicle maturation. Okay, this is what's happening. And then, of course, once the follicle is mature, that's when ovulation will occur. And then after that, we're going to have the luteal phase, which I'll talk about. Okay? Okay, so this is driving the first half of the cycle here, this maturation process. So what ends up happening is, as this secondary follicle, remember the secondary follicle now is the one that has, go back a couple screens, we're down here. Okay? We've got an enlarged primary oocyte. One of those was selected, and whenever one started to get larger, dividing, and you've got a secondary oocyte, and that's where we're at in this picture here. Okay? What is happening? Notice Notice what's making this entire follicle larger. The actual granulosal cells that are around the outside just keep compounding, and they keep getting larger, and they make this follicle larger. Okay? And what you will see is the signals of the cells. There's connective tissue, right, all through the ovary that's around all of these. The cells that are around here receive a chemical message and start getting converted into what's called thecal cells, Thecal. They're going to be important here in a little bit. Okay. Now, it says during the follicular phase, the granulosal cells of some primary follicles proliferate. Remember, only a handful of them are actually going to get called up to the big leagues. Okay. And the oocyte itself continues to enlarge as well during this process. Now, this is showing one, but there can be more than one happening. Now, then what will start to happen is, if you take a look from the previous screen, where the density of these cells, and look at the next screen, notice what's happening. Cavities are starting to form. Now, this is important, okay? It's going to allow it to get bigger and bigger and bigger and take up more space. We'll call that space in there the antrum, okay? And the antrum is going to continue to enlarge in this secondary follicle. And then what starts to happen is, when it gets big enough, like this, these thecal cells, as they increase in number, I was talking about, these thecal cells on the outside, they start to secrete estrogen. Okay? Whenever these cells start to secrete estrogen, it ups the level of estrogen in your body. So from day 0 to, let's say, day 14, your estrogen levels are climbing. And the reason that they are climbing is because during this follicular development, as you are moving into the formation of a secondary oocyte, more of these thecal cells are starting to form around the outside of that follicle. More of those cells means more estrogen. Okay? So the estrogen levels start to rise. What's going to happen when they reach their peak is the question. All right, so notice now, look, just prior to ovulation, look how much space the antrum has taken up. Okay? So this is still the entire follicle. All this large amount of, notice this is 12 to 16 millimeters. You know, that's reasonably large, right? There's 10 millimeters in a centimeter, right? So this is, anywhere from 1 to 2 centimeters big. There's 2.54 centimeters in an inch. So, this is taking up a considerable amount of space when this happens. Okay. Now, what will happen is this will then start to press on and fuse with the ovarian surface. Okay, and eventually this will lead to an eruption and actually a reasonably violent explosion of an egg. Okay. So when that happens, there you go. Notice what goes out with it. Some of these granulosal cells, right? It's still around it, still with it. You've got a follicular remnant here, and what's going to happen as a result of this is this is going to turn into something else. So if this is midway through and this happens, now what's going to take over? Because the follicle isn't developing anymore, so you're not increasing the amount of these thecal cells that are going to increase estrogen levels. Instead, you're going to start going back the other way. And what's going to happen is these cells are all going to convert into a different sort of form of lump of endocrine-type cells. They're going to start secreting, well, it depends on if this gets fertilized and impregnated or not, but depending on what happens to this changes and secretes another hormone, okay, that's going to take you back the other way. Keep in mind as we'll get to fertilization in a minute, but this may or may not be fertilized, right? It's going to be released into the oviduct. Don't know which side. It could be right, could be left, could be both, could be none. Just depends, but that's where it could go. Okay, so we know this is actually the ovulation that is happening here. All right, now this is where we start the luteal phase. So up until this point, we went from day 0 to let's say day 14. We've got to ovulation. Up until that point, estrogen levels were rising. There's changes associated with the body when estrogen levels are rising. There's mucosal changes that are apparent, outwardly. There's a lot of other things that are happening. Okay, and we'll get to some of those a little bit later, I think. But at this point, we're starting the luteal phase. It's called the luteal phase because of we've got luteinizing hormone, right? LH. Up until this point, FSH levels were increasing. Follicle-stimulating hormone, because we were stimulating that follicle. We were growing that follicle. Well, we weren't. You were. Now, at this point, we've got what's called the corpus luteum that's behind. You'll see that it almost like it turns to fat, but it's not fat. It's endocrine tissue. Okay? This degenerates what was left of that follicle and turns into something else, and then we've got a change now. in direction of the cycle. This thing continues to mature, continues to develop, and if the egg gets fertilized and implants, then what will happen is that will continue, that corpus luteum will stay around and secrete progesterone and help nourish that environment and make it better for developing embryo, whatever. If it does not, if a woman does not get pregnant at this point, this continues to degenerate, and when it degenerates, its hormone levels dwindle as well, what it's secreting. So it falls back down basically until you get to the start again, and then we start this whole thing over. Okay? And I'll show you the hormonal levels here in a second of actual graph. Yes. When the embryo implants and there's something when you become pregnant, the corpus luteum will stick around and it will continue to secrete progesterone to help with that. It will not if you do not become pregnant. It will degenerate. You mean if not? Yeah, it just degenerates. Its parts get reabsorbed. It's just like anything else. Like when a cell destroys something and takes it away, the same thing will happen. Okay, so like I said, the corpus luteum operates the last 14 days. All right. It's the converted follicle. I told you that already. I told you it's going to increase the amount of progesterone. Now, progesterone was already being released. Don't think that it wasn't. The fact that luteinizing hormone was already climbing a little bit because of the signal from gonadotropic releasing hormone from the hypothalamus. LH was rising a little bit already, as was FSH. But you're going to get a big spike and a big jump in progesterone levels if that corpus luteum stays around. It's going to be the difference between the progesterone levels of a pregnant woman and the progesterone levels of a non-pregnant woman. Okay. So it's going to degenerate about 14 days. It'll be done degenerating if after its formation, unless fertilization and implantation occurs. Like I said, if it does occur, it's going to stick around and give its hormones. It's now called the corpus luteum of pregnancy. I want to show you this. Won't these ever work? Here we go. All right, here we go. Ridiculous. Oh, here we go.
At sexual maturity, each of a woman's ovaries contains about 200,000 immature eggs called primary oocytes. A primary oocyte is diploid and is arrested in prophase 1 of meiosis. A layer of follicle cells surrounds each primary oocyte. Together, an oocyte and its follicle cells make up a follicle. An ovarian cycle lasts about 28 days, beginning at the first day of menstruation, or menses. During the first 7 days of the cycle, 6 to 12 primary oocytes begin to mature. As the follicles develop, the follicle cells communicate with oocytes and pass them nutrients through pores called gap junctions. Each oocyte grows larger, and the surrounding follicle cells divide, proliferating to produce thousands of follicle cells in a single follicle. By day 7, all but one of the developing follicles begins to degenerate. The remaining follicle continues to develop, and its follicle cells continue to pump it with nourishment, and also supply it with
controls
proteins
that, you know?
and informational
Who
molecules
gets to decide who
needed
wins?
for early stages
I
of
don't know.
development. The maturing primary oocyte completes meiosis 1 and divides into 2 haploid cells. Each of these cells receives half the chromosomes. However, one cell, called a polar body, receives very little cytoplasm. The other, now a secondary oocyte, enters meiosis 2 and rests there until fertilization. At day 14, ovulation occurs, and the secondary oocyte erupts from the ovary. The oviduct contains microscopic cilia that beat and draw in the released oocytes. This immature egg enters an oviduct, where it may become fertilized by a sperm cell and complete meiosis. The follicle cells that are left behind develop into a small mass of endocrine tissue called the corpus luteum. The corpus luteum remains in the ovary for 2 weeks, secreting the hormones estrogen and progesterone. At the end of the ovarian cycle, if the woman is not pregnant, the corpus luteum disintegrates. Each month, the ovarian cycle is tightly coordinated with the uterine cycle. In the uterine cycle, the lining of the uterus builds up, forming elongated glands, and then sloughs off. The cycle begins with the sloughing of the uterine lining. This is the first day of menses, also called menstruation. After menstruation, the uterine lining starts to grow again and to prepare for implantation of an embryo. During this phase of the uterine cycle, up until ovulation, the uterine lining proliferates, and the glands that make up the uterine lining elongate. Capillary beds grow into the glands to supply this tissue with nutrients. Just before menstruation occurs again, the capillary beds degenerate and no longer deliver the surrounding glandular tissue with nutrients. During menstruation, this tissue dies and sloughs off through the vagina to the outside of the body.
Okay, now, here's the complicated part. We're going to take a look at the overlaying hormone levels and how that drives all of this. Okay. If you take a look, I'm going to focus, down here is the corresponding, what we have set up here is, here's the days across here. Okay. This would be endometrial thickness. We're starting at the beginning with it sloughing off, decreasing in thickness. Remember it said the capillary beds pull back from that glandular tissue and it starts to disintegrate, and that comes out with during menstruation. But when that decreases, you're starting over again, and we've got a gradual building of the uterine lining until it starts again. So that's where we're starting. This is our starting point. This is showing what is happening in the ovary in terms of the individual follicles that correspond at the same timeline. Okay, that's what I like about this. It's a timeline showing everything that's happening. This is going to be looking at the actual low-level hormones, the estrogen and progesterone. How are those levels changing in response to the high-level hormones, FSH and LH? Okay, so let's take a look at this and see if we can make some sense out of it. During the follicular phase, it's the beginning. This is number 1 and number 2. So here's number 2. Number 1 is just in reference to the days that you can see where we're at. Okay, so anywhere up through the first couple weeks or so is the follicular phase. So we're starting right here. If you look at the follicle, we're moving from a primary oocyte into the development of a sort of semi-mature secondary oocyte at this point. Okay, so this is where we are. A rise in follicle-stimulating hormone signals the ovarian follicle, or the thecal cells therein, to secrete more estrogen. Now remember what's happening. As this guy is developing, look at those cells that are around it. More of those cells, the connective tissue cells around this follicle, become thecal cells, and they start to, as they are made, start to secrete more estrogen. Now where does that signal come from? It comes from the fact that FSH levels, notice it's a small hump, but it's a little one nonetheless. It's coming up and it's rising, and it rises enough to increase these thecal cell production, and then they will contribute also more to actual estrogen production. Okay? So what you'll see is a slow increase in estrogen as a result of that. All right, so a rise in FSH, you can can see that right here. Starts to come up a little bit. Signals the ovarian follicle, this guy down here, to secrete more estrogen, which is what we're seeing right here. Notice that as about when this starts to go down is at that point where the feedback was happening, and that's where about this starts to go up. Okay, so we can correlate these two hormones together with one another. So a rise in estrogen feeds back to inhibit FSH. So that's why this continues, as this continues to rise, the estrogen continues to rise. What was calling on that, what was coding for that, I guess, is FSH. See that it continues to go down. As estrogen goes up, it goes down because that's a negative feedback cycle. All right, so FSH was the key. It's delivering that. It's helping the follicle make more Thickle cells, which is going to produce more estrogen, and that's why you're going to see a rise in estrogen. And as that estrogen continues to rise, it negatively feeds back and shuts off this FSH, okay, or at least slows it down. All right, so it says, excuse me, what will happen is to inhibit FSH secretion, which declines during as the follicular phase proceeds, and it continues to decline. I just said that. All right, LH rises, luteinizing hormone, okay. Luteinizing hormone rises in the follicular phase, so let's look at 5 and 7. We see that LH is rising. Look how much more linear it is rising. There's no interplay of it right now. There doesn't seem to be, it's just rising for the sake of rising because it's not having a direct impact at this point, but it's getting larger and larger, okay. It's going up, 5, 7, you see there, and as it peaks in mid-cycle, which is right here, you see it shoot up. It gets really high right during this mid-cycle, okay. Luteinizing hormone spikes. It triggers at that point ovulation because what will happen is you will see how that is correlated with the spike in estrogen as well. You'll see that as these, sort of like what we talked about back with neurons, there was a threshold, right, like voltage-gated channels and that kind of stuff. There's like certain levels that once they are reached, things just sort of all open up and something happens. You got another situation like that here, okay? So as these estrogen levels continue to rise, these are rising accordingly until a certain level is reached and it causes those to spike. So you get a release of luteinizing hormone. Okay, so here's ovulation. Let's look and see where we're at. Down here, we've got a release of the egg. The second, here's what the uterus looks like, the endometrium, I guess, at this point. We're constantly thickening. This is mostly contributing to that. We've got estrogen. This is triggering this estrogen. This increased level of estrogen is feeding back down here. A little bit of progesterone is feeding into this as well, and it's building this lining, okay? And we're at about right here, somewhere in here. Not quite as full as it's going to be. Okay, notice what happens, number 8, at estrogen as soon as this ovulation occurs. We know that the cells that were most responsible for that surge in estrogen are now no longer there. The follicle is open and the egg is out, and now these are not functioning anymore. So what do you get is a very steep drop in estrogen levels right at that point. You can keep track of all this, right? And there's, you can certainly keep track of it with blood tests, but you could also see that there's bodily changes that are associated with these hormone levels as well. So it's temperature changes, mucosal secretions, all kinds of stuff that accompany these changes. So you get a plummet in the estrogen level. Okay, so that's estrogen output decreases. The mature follicle is converted to a corpus luteum. So see that? That's what we got left over. Okay, so what's happening here? We're still thickening down here. We've got a released follicle, an ovulated egg at this point. We've got a leftover corpus luteum. So what happens now? Notice what is happening here. Look at the progesterone levels. They work toward a flat line for a while. Why do they go up? Because this guy starts to produce a lot of progesterone, this corpus luteum. You'll see that those levels start to rise. But not only progesterone, that's the key. I mentioned progesterone with this because that's what you see the biggest change in. Because up until that point, you basically don't see anything with it. You're also going to get an estrogen release from this. Okay, both hormones are involved. Progesterone mainly, it spikes the highest, but you'll see that both of those increase right around this time. You can see that. Okay, now as both of those increase, you get a big, sort of how that kind of plateaued, leveled off, and then all of a sudden you got a big growth right here. That corresponds to this fluctuation right here. Now all of a sudden you've got substantial thickening of this endometrium. All right. Now what will happen is, notice that this is negative feedback. Progesterone and estrogen right here, as they continue to rise, shut this down. So this falls because these start to rise. And that's a negative feedback. Okay? All right. So that's this. Progesterone output inhibits the release of FSH, so there's 16. This increases, this decreases as a result. And luteinizing hormone. With low luteinizing hormone, the corpus luteum degenerates. This is assuming that pregnancy doesn't occur. Okay? So as this starts to decrease, this is increasing, right? The progesterone is increasing, this will start to decreasing, and eventually this is going to lead to a breakdown in terms of this corpus luteum over here. All right. Now, what can happen at this point? If the corpus luteum is broken down and it disintegrates and it's not there to maintain this high level, as long as this level right here is maintained, okay, this will stay down because that's a negative feedback cycle. But the second this starts to degenerate and this starts to decrease, guess what happens? You're not getting that negative feedback, so what happens? Now we've come cycled. We've come back around, because if this falls, guess what's going to climb? FSH and LH all over again. Now we've come full circle, and it's going to do it all again. And it takes about 28 days on average for all this cycle to work itself out. So it's pretty interesting, fairly complex, but kind of cool. Now we can manipulate all of this. We'll talk about some birth control stuff a little bit later, and then how we can affect these levels. But okay. Now let's look at a better picture of what I just said a second ago. This works. Why is this so slow?
At sexual maturity.
Nah, we already said that one. Sorry.
The ovarian and uterine cycle.
Here we go.
The ovarian cycle is controlled by the interplay of hormones from the pituitary gland and from the ovary itself. A few days before the beginning of the cycle, the anterior pituitary begins to increase its secretion of two hormones, follicle-stimulating hormone,
So this is
FSH,
increasing.
and luteinizing hormone, LH. FSH and LH stimulate ovarian follicles to grow. As the follicles grow, they begin to secrete estrogen. During this phase of the cycle, the increasing levels of estrogen feed back on the pituitary to inhibit the release of additional FSH
So that's
and
why
LH.
we see a decrease here.
During the next week, the levels of FSH and LH drop. Beginning around day 12, the increasing levels of estrogen suddenly have the opposite effect on the pituitary gland. Instead of exerting a negative feedback on the pituitary, these hormones now exert a positive feedback, stimulating the pituitary to release FSH and large amounts of LH. LH reaches a peak at day 14 of the ovarian cycle. This LH surge triggers the mature follicle to rupture and release the egg, the process of ovulation. LH then triggers the remaining follicle cells to differentiate into the corpus luteum, which secretes estrogen and progesterone. The corpus luteum remains in the ovary, secreting estrogen and progesterone for the last 2 weeks of the cycle. At this point in the cycle, these hormones again inhibit the release of FSH and LH. A decline in FSH and LH restricts follicles from beginning to develop during the second half of the cycle. LH, or a hormone produced by an implanted embryo, is required to maintain the life of the corpus luteum. At the end of the cycle, if an embryo is not implanted, the corpus luteum degenerates. When the corpus luteum degenerates, it no longer releases estrogen and progesterone.
One thing that was interesting, I don't know if you picked up on this or not, but as long as these levels stay low and they don't climb, you're not going to initiate the development of another follicle. So, the reason, why is it that you can have ovulation right here, and why can't you have another quick ovulation right behind that? Well, because these hormone levels, this is what triggered ovulation. So unless that is high enough, as long as this is here, as long as the corpus luteum is present, it is secreting these hormones. And by virtue of the fact that these hormones are there and secreted because that guy's there, these stay down. As long as these stay down, you're not getting another one until this process is over. So that's kind of interesting. All right.
The ovarian,
See if this is different. I think this is.
the ovarian and uterine cycles are tightly coordinated. Hormones secreted by the ovary at different phases of the ovarian cycle trigger changes in the uterine lining. For example, at the beginning of the cycles, the levels of estrogen and progesterone are too low to maintain the uterine lining, and menses begins. About a week into the ovarian cycle, the developing follicle increases its secretion of estrogen, and estrogen levels in the body begin to rise. This hormone triggers the cells of the uterine lining to proliferate, and the lining becomes thicker. Just before ovulation, the level of estrogen in the body has reached its peak. Afterward, the follicle cells remaining in the ovary develop into the corpus luteum, a structure that releases estrogen and progesterone. The hormones maintain the uterine lining at a peak thickness and preparedness for embryo implantation. At the end of the cycle, if the egg is not fertilized or has not implanted, the corpus luteum breaks down and stops releasing estrogen and progesterone. Without these hormones, the uterine lining also breaks down, initiating menses.
So that pretty much covered this right here. So let's go ahead and skip this slide here. One thing here. I mentioned this already, but when you fluctuate those estrogen and progesterone levels, it does produce changes in cervical mucus. So that's one way to track it. There's also some temperature changes associated with the body. You guys probably know all of that already. As far as the pubertal changes that happen in females, it's similar to males in the sense that obviously gonadotropin-releasing hormone is the key. That's the one from the hypothalamus. That's the tropic hormone. And it's going to stimulate FSH, that's supposed to be FSH, not FHS. FSH and LH, just like it did in males, those anterior pituitary hormones. But in this case, the estrogen release is what's going to be most important for the female. A lot of changes are going to occur. Reproductive system will mature. All that other stuff that goes with it. Now, some may argue this, but menopause is unique in females. Now, what I mean by menopause is a well-described physiological set of conditions that is reasonably well studied. Some people will say the whole male midlife crisis, male menopause thing. I'm not saying that that's non-existent. I'm just saying from a research standpoint, not as well defined. Okay, so by definition, we're going to say that this is unique in females because we're looking at hormonal, the actual estrogen, progesterone, that kind of stuff. So what happens is, and this is neat because it's a mystery in terms of what controls this. What are the factors that go into initiating this change? What biochemically is happening? It's pretty interesting stuff. Something happens in the hypothalamus. Okay, there's a midlife hypothalamic change. This whole feedback mechanism, it could happen primarily, it could happen secondarily, it could happen way up at the top, at the very top tropic hormone. Something happens, and it probably starts with the hypothalamus. That's what is suggested. And somewhere along the line, during all these different mechanisms, we've talked about how it feeds back, and this produces this, and so on and so forth. That slows down, and the synchronicity of it all gets overlapped or delayed. And as a result, the cycles become irregular because they're not as predictable as normally how well they were controlled. So you get increasingly irregular cycles down to the point where it could be, dwindling estrogen levels, worst case scenario, lack of estrogen levels. And of course, it's obvious that this could cause extremely important physiological actions and reactions. There's a lot, you cannot change hormonal levels and to the degree that all of this is involved with in the body and not expect a behavioral change, a physiological change. But anyways, now I want to mention this before we go because this is the fun part. And this is where it gets ethically gray. We'll talk about some bioethics here as we do this. Fertilization happens way up here. Okay, it doesn't happen way down here. Now, implantation is not supposed to happen up here. It's supposed to happen down here. But what is some points of interest here. Okay, the upper third of the tube is where they should get fertilized. So think of how long and how far those sperm must have traveled to get to that point. Okay, way up here. Notice some of them just took a wrong turn. They're going the wrong way. Notice visually, do what?
They didn't stop and ask for directions.
Huh. I like that. I like that. All right. These poor people. These poor people listen to this. I'm coughing. I'm clearing my throat. My voice is hoarse. I got to put up with this.
You do put your hand up there a lot. Is that to cover the mic?
No, it's because my throat hurts. I forgot it in my office. That's my problem. Okay. I'm all right. Thanks. Anyways, visually take a visual reference. If you look at the concentration of the sperm down here versus what makes it way up here, there's some numbers over here we'll mention, but you can see there's a big difference in terms of where you started versus where you ended. But fertilization must occur within 24 hours after ovulation. You got a 24-hour window. Now, that does not mean that you can only have sex prior to that window, and that's how much time you got, because sperm actually can survive up to 5 days. Actually, it's been shown anywhere from 3 to 7 days in the female reproductive tract. So ejaculated sperm from a week ago could actually still be viable, okay? But the point of fertilization, that egg is viable for 24 hours, is what that's saying. Okay? So you can play with the numbers here and figure out how much room you got to work with. But of course, a tubal pregnancy is also called, it's an ectopic pregnancy. That's what this is. Okay, so it gets implanted up here. And we all know that this lining is not designed to get thickened and nourished like this lining is down here. So you've got two big problems. Number one, the baby's not going to develop down there. And secondly, this could cause a lot of harm to the mother. So, this is interesting because it puts in, ethically speaking, it puts people in some tough situations sometimes, depending on how you believe about certain things, and depending on how far down. I mean, are you right here? Are you back here? I mean, it's hard to come up with things unless you've experienced them. But well, you can, and that gets great too, because who wants to try to do that? I mean, if they do that and you're unsuccessful, I mean, have you or have you not? You know. Yeah, right. So I mean, well, it's not just religion either. I mean, a lot of it could be, but a lot of it is just straight up science ethics as well. I mean, at what point should we intervene? At what point are we working against? Let's take the faith-based thing out of everything. At what point are we working against the laws of nature that science has shown us that occur? And that kind of stuff. I know it's kind of gray, but anyways. Go ahead.
But the woman's, she had an ectopic pregnancy, and it ruptured, and the baby survived and implanted onto one of her organs. And she carried it for like 2 months before term, and they went in and took it.
Mm-hmm.
And then she had twins in her actual uterus, so it was like she had triplets.
At the same time? Oh, that would suck. That's crazy. I've never heard of that. But
The ruptured fallopian tube, she's like, you didn't kill her.
Yeah.
Yeah, because usually that's what happens.
Huh. That's really neat. Huh. Well, let me finish this slide real quick. We know the path. You got to go past the cervical canal, all that, to get up here and to get into the tube. But sperm migration is actually helped by the female tract. There's actually contractions of the myometrium, the actual muscle of the uterus. And remember, prostaglandins initiate some of this contraction. I mentioned that before. There's actually a propelled sort of unidirectional movement to help migrate these sperm where they need to go. And of course, they're flapping like crazy to get there as well. Acrosomal enzymes are, I remember, around the head of that sperm that are going to, when they get there, enable them to digest their way into that egg. Okay. And here's what's really neat about this whole communication process. I said I was going to mention something a while ago about there's actually, during this time, and the chemotaxis, the chemical drawing of the sperm to the egg, there's been, the last time I went to a conference a few years ago, and a guy was speaking about this. I don't know what they found since then. But lily of the valley, it's an actual scent. That chemical could be used, I mean, maybe it's a pheromone. In this sense, it would be by definition. Has been isolated from this tract, in this area, before and after. So in other words, it wasn't there, and then all of a sudden that chemical comes into play. Does that have a bearing? Since it's not there before and it's there now, is there a pheromone? We know that there's a chemical attraction. We find it appealing at some level. Is that some sort of communication between egg and sperm? And if so, that sheds a whole new light on terms of buying flowers for people. Right? So, but that's not a joke. That's actually real. That was an actual chemical that they've isolated from this. Anyways, the last thing I want to end with is this picture down here. Oh, and what I was going to say is there are mechanisms. Two sperm will not fertilize the same egg. If you have twins or whatever, you know that that's egg division, right? The sperm, no two things in this universe occur at the exact same time. So even if they're really, really close and it's a tie, one of them is by virtue of definition of all the laws of physics, one of them is going to actually be first. So if that's the case, the egg will shut down and go through a bunch of biochemical changes to prevent more than one sperm ever fertilizing an egg. If ever somehow that happens and the chromosomal number becomes different, generally you get spontaneous abortion, or else you don't get fertilization, one of the two. But anyway, I thought that was neat. And then here's numbers that correspond with this picture. Percent of ejaculated sperm, obviously 100% of it or close to it can actually find its way in to the vagina. But notice that by the time you get to the actual cervical canal, look, you've lost 97% of the sperm. Okay? You've actually only got 3% of the sperm actually make it even through the cervical canal. Now, into the uterus, once you get to the uterus, look, 0.1%. I'm not talking about 10%, 0.1. I'm talking about 0.1%. So what's 0.001? That's a very small amount compared to what you started with. And then actually at the fertilization site, which is about 30 to 60 minutes later, notice here's the time frame, 1 to 3 minutes, 10 to 20 minutes, 30 to 60 minutes, 0.001%. So you've lost a bunch at that point. So, all right, we'll end there.